All right, let's unpack this. When you hear the word cholesterol, what immediately springs to mind? Is it the friend, an absolutely essential component of every single cell in your body? Or is it, the notorious villain lurking in your arteries, threatening your heart health?
Yeah, it's got that reputation, doesn't it?
For most of us, it's a word we hear constantly in health news, diet discussions, medical checkups, often triggering maybe a subtle shiver of dread. But how many of us truly understand what it is, what it does, or the intricate dance our bodies perform to manage it?
It's quite remarkable how a single molecule can carry such a dual reputation, isn't it? We hear high cholesterol and often immediately jump to bad. But the reality, as we'll explore today, is far richer and more nuanced.
I couldn't agree more. That's precisely why we're here. Welcome to the Deep Dive, where today we're not just scratching the surface of cholesterol. We are taking a truly immersive journey into its complex, vital, and yes, sometimes perilous world. Our mission for you, our listener, is to gain a mastery-level understanding of this crucial lipid. We're going to cover everything mentioned in our sources, diving deep into every concept, because we believe that to truly be well-informed, you need to grasp the foundational science.
We aim to equip you with insights usually reserved for professionals in the field, helping you connect the biochemical dots to real-world health implications. This isn't about rote memorization. It's about understanding the why behind the what and, appreciating the body's incredible design.
To guide us through this intricate landscape, we've taken a stack of expert sources, including core biochemistry texts, and distilled their essence. We've done the deep diving for you so you can focus on absorbing the critical insights.
They'll be your guides,
helping you navigate the biochemical pathways and clinical implications, ensuring you walk away truly well-informed.
So how will we navigate this complex world today? Where do we start?
We'll begin by truly appreciating cholesterol's dual nature, how it's absolutely essential for life, yet undeniably capable of causing serious disease. From there, we'll take a microscopic journey into your cells to witness its meticulous creation from surprisingly simple building blocks.
And once it's made, how does this vital molecule travel? We'll then track its fascinating routes through your bloodstream, exploring the roles of the various lipoproteins, the LDLs and HDLs that everyone talks about, and then some.
Yeah, exactly. Finally, we'll uncover how your body gets rid of cholesterol, connecting all of this to real-world health implications, from the food you eat and the lifestyle you lead, to cutting-edge drug therapies and even inherited conditions that can throw your cholesterol balance completely off kilter.
Sounds like a thorough plan.
So let's begin by truly appreciating cholesterol for what it is. An unsung hero and sometimes a notorious villain.
Ooh.
Let's kick things off by appreciating cholesterol's profound biomedical importance. It's truly so much more than just a bad reputation, isn't it?
Oh, absolutely.
We often hear it as a negative, but it's fundamentally essential for us to even be alive.
That's a crucial point. Cholesterol is an amphipathic lipid. Now, what this means is that it has a split personality, basically. One part loves water, it's hydrophilic, and the other part hates water, it's hydrophobic.
Okay.
This unique dual nature is absolutely critical to its function, particularly its role as an essential structural component of all our cell membranes.
So because it's amphipathic, it can comfortably embed itself right within the fatty bilayer of our cell membranes.
Exactly.
The sources highlight its role there as being vital for maintaining the correct permeability and fluidity of these membranes. Why is that specific function so incredibly important for every single cell in our body?
Well, imagine your cell membrane like the walls of a flexible living house. If those walls become too rigid, nothing can get in or out, and the house becomes brittle.
Right.
If they become too flimsy, the house loses its shape and integrity, spilling its contents. Cholesterol acts like a molecular thermostat or maybe a buffer for that membrane. At higher temperatures, it actually reduces fluidity, preventing the membrane from becoming overly leaky or fluid. At lower temperatures, it prevents the membrane from solidifying, ensuring it maintains essential flexibility.
So it keeps things just right.
Precisely. This precise control over permeability and fluidity is fundamental for virtually every cellular process, from nutrient transport into the cell to the precise transmission of nerve impulses. Without cholesterol, our cells simply couldn't function properly.
That's a fantastic analogy. It makes so much sense. And it's not just in the basic structure of cell membranes, is it? The sources mentioned it's also a key component in the outer layer of plasma lipoproteins.
Yes, those transport vehicles we'll talk more about later.
Got it.
But beyond its structural role, what's truly foundational about cholesterol is its role as the precursor of all other steroids in the body. It's like the master template from which countless other vital molecules are built.
The starting point.
Exactly. Without it, entire hormonal systems wouldn't exist.
That's fascinating. When we say all other steroids, we're talking about an incredibly diverse and powerful array of molecules. For instance, tell us about corticosteroids.
Right. Corticosteroids are crucial hormones involved in your body's stress response, regulating metabolism, and modulating inflammation. Think about cortisol, often called the stress hormone. It's directly derived from cholesterol.
Wow. And it's not just stress, right? What about things like reproduction and development?
Absolutely. Then there are the sex hormones, testosterone, estrogen, and progesterone. These are fundamental for reproductive health, sexual development, and numerous physiological processes in both men and women.
All from cholesterol.
Cholesterol provides the basic skeleton for all of them.
And it doesn't stop there. Cholesterol is also the precursor to bile acids, which, we'll dive into deeper later. But for now, suffice it to say, they are absolutely essential for fat digestion and absorption in your gut.
Critical for getting nutrients.
If you can't digest fats properly, you can't absorb essential fat-soluble vitamins or get vital energy.
And of course, vitamin D. This isn't just for bone health, though that's important. It's actually a critical hormone that plays widespread roles in immune function, cell growth, and releasing inflammation.
And that starts with cholesterol, too.
It all begins with the cholesterol precursor that gets modified by sunlight on your skin. So, you see, cholesterol is far from just a bad molecule. It's a linchpin for life itself.
Okay, so we've established its vital roles. Now, where does this cholesterol actually come from? Is it something we just eat, or does our body make it?
Well, here's another surprising fact for many. Over half of the body's cholesterol, approximately 700 milligrams per day, is actually synthesized internally.
Roughly over half.
Yeah, this is what we call endogenous synthesis. Your body is incredibly self-sufficient in this regard.
So our bodies are literally little cholesterol factories. Which specific parts of the body are doing the heavy lifting in this regard?
Well, virtually all nucleated cells in your body are capable of synthesizing cholesterol if they need it, but the liver and the intestine are the primary production hubs. Each accounts for about 10% of the total synthesis in humans.
Okay, liver and intestine.
And this complex synthesis process occurs within specific cellular compartments. The endoplasmic reticulum and the cytosolic compartments, basically the internal machinery of the cell.
So if over half is made internally, the remainder must come from our diet, the exogenous sources. This is where cholesterol often gets its negative reputation, right?
Indeed. Dietary cholesterol is a product of animal metabolism. So foods like egg yolk, meat, liver, and brain are typical sources.
The animal products.
Right. It's an important distinction that plants don't produce cholesterol, though some plant sterols can impact cholesterol absorption, which is a whole other thing.
Interesting. Okay, so we've got this essential molecule, vital for so many functions, but then there's its dark side. Let's talk about the pathologic dual nature, when this usually good cholesterol goes bad.
Yeah. The sources mention two primary pathological roles. First, it's a major constituent of gallstones. These are hardened deposits that can form in your gallbladder, leading to pain and other complications.
Ouch.
And then its chief pathologic role, the one that truly dominates health discussions, its contribution to the development of atherosclerosis.
Right, the hardening of the arteries.
This isn't just a minor issue, it's a foundational process for major diseases that affect millions.
Which one specifically?
Atherosclerosis is the underlying cause of cerebrovascular disease, which can lead to strokes, coronary heart disease, which leads to heart attacks, and peripheral vascular disease affecting blood flow to the limbs.
Wow, okay.
Understanding this connection is absolutely key, and we'll delve much deeper into the mechanics of atherosclerosis later. But for now, just know that cholesterol is a central, pivotal player in this potentially devastating process.
That sets the stage beautifully. Now let's get truly granular. We're moving from the big picture to the intricate blueprint, the biosynthesis of cholesterol from acetyl-CoA.
Wow.
This is where we see the magic happening inside your cells, how something so complex is built from such simple beginnings.
This really is a remarkable feat of molecular engineering. Every single one of the 27 carbon atoms that make up a molecule of cholesterol is derived from just one starting block, acetyl-CoA.
Just acetyl-CoA.
That's it. Think about that. A relatively simple 2-carbon unit is transformed into this complex 27-carbon compound, which has a distinct 4-ring structure and a side chain. It's like building a grand cathedral from countless identical bricks.
And the sources break this intricate process down into 5 major conceptual steps, each one a symphony of enzyme activity. Let's start with the first movement, the initial construction of mevalonate. How does our body begin this construction project?
Okay, so this initial step is essentially about carefully linking those 2-carbon acetyl-CoA Lego bricks together. 2 acetyl-CoAs condense, then a third joins in, forming an intermediate called HMG-CoA.
HMG-CoA.
And here's where it gets really interesting. This HMG-CoA is then reduced to mevalonate in a reaction catalyzed by a very special enzyme called HMG-CoA reductase.
HMG-CoA reductase. Okay, that name sounds familiar. This isn't just any enzyme, is it?
Absolutely not. The sources highlight this as the principal regulatory step in the entire cholesterol synthesis pathway. Think of it as the primary choke point, the master switch, or, the faucet controlling the flow.
So the main control point.
Because of this, HMG-CoA reductase is the direct target for the most effective class of cholesterol-lowering drugs on the market, the statins.
Ah, that's where they work.
Exactly. Inhibiting this specific enzyme is precisely how those drugs prevent your body from making too much cholesterol.
Fascinating. So that first step is absolutely crucial for both natural regulation and drug intervention. What happens next after we have mevalonate?
Step 2 involves transforming a mevalonate into active 5-carbon isoprenoid units. Mevalonate undergoes a series of careful modifications involving phosphorylation, adding phosphate groups using cellular energy, ATP.
Right, adding energy.
And then a decarboxylation where a small carbon dioxide molecule is removed. The result is the active 5-carbon building block called isopentenyl diphosphate.
Isopentenyl diphosphate.
Okay. This little 5-carbon unit is the fundamental building block from which the larger ringed cholesterol molecule will be constructed.
So we've got these small, activated 5-carbon units. How do they start coming together to form something as large as squalene? That's step 3, the condensation of 6 of these isoprenoid units.
Think of it as a precise molecular assembly line. First, some of the isopentenyl diphosphate units are isomerized, essentially reorganizing themselves slightly to create a partner molecule called dimethylallyl diphosphate.
Okay.
Then, these 5-carbon units begin a series of precise snap-together reactions. A 5-carbon and another 5-carbon unit combine to form a 10-carbon molecule.
Geranyl diphosphate.
That's it, geranyl diphosphate. Then that 10-carbon molecule grabs another 5-carbon unit to form a 15-carbon piece called farnesyl diphosphate.
So we're building it up piece by piece, 5 carbons at a time. What's the grand finale of this phase?
The grand finale of this phase is when 2 of those 15-carbon farnesyl diphosphate pieces finally link up head-to-head, forming the large 30-carbon compound called squalene.
Squalene, 30 carbons.
This process involves eliminating some molecular waste pyrophosphate and adding energy via NADPH, catalyzed by squalene synthetase. It's like constructing the full long skeleton before adding the protective layers or, closing the rings.
That's quite a complex series of condensations. And the sources note that farnesyl diphosphate isn't just for cholesterol. It's an intermediate for other important compounds, too. What are some of those?
That's a great observation about the efficiency of biochemical pathways. Farnesyl diphosphate is a precursor for dolichol, which is crucial for protein glycosylation. That's a process where sugar chains are added to proteins, impacting their function and targeting.
Okay.
It also leads to ubiquinone, also known as coenzyme Q, a vital component of the electron transport chain, essential for cellular energy production.
CoQ10, right.
That's the one. And there's also the mention of protein prenylation.
What's that about?
Protein prenylation involves attaching these same farnesyl or geranyl residues, which are 15 and 20 carbon isoprenoids, respectively to certain proteins, particularly those involved in cell signaling, like RAS proteins.
Okay, so attaching fatty tails.
Essentially, yes. This modification helps anchor these proteins into cell membranes and is believed to be involved in crucial protein interactions and the precise trafficking of membrane-associated proteins within the cell. It's another example of how central these isoprenoid intermediates are to broader cellular functions beyond just cholesterol.
Truly intricate. So we've got squalene, a 30-carbon open-chain molecule. The next step, step 4, is where the rings form. The cyclization of squalene to form linosterol. How does an open chain become a complex ringed structure?
Squalene has this remarkable innate ability to fold into a very specific 3D structure that closely resembles the steroid nucleus. Before the rings actually close, squalene is subtly modified by squalene epoxidase, adding an oxygen atom. Then, with the help of an enzyme called oxidosqualene linosterol cyclase, an almost magical rearrangement occurs. Internal bonds shift, and methyl groups migrate, leading to the rapid closure of the 4 rings.
All in one go.
Pretty much. This precise, single-step reaction forms linosterol, which is considered the parent steroid, the very first true sterol in the pathway.
And finally, step 5, the formation of cholesterol from linosterol. We're so close to the final product. What modifications happen here?
This final series of transformations also occurs in the membranes of the endoplasmic reticulum. It involves multiple precise steps. Essentially, some excess methyl groups are removed from specific carbons, C14 and C4.
Cleaning it up.
Exactly. Then double bonds within the molecule shift to their final positions, like moving from C8-C9 to C5-C6. And finally, a double bond in the side chain is reduced. After these final refinements, you finally yield the complex yet elegantly structured end product, cholesterol.
Wow. So if you were to distill all that incredibly detailed molecular architecture down, what's the most profound insight that process reveals about our bodies for us, the listeners?
It's this. Your body is a meticulously organized chemical factory, capable of building a complex, vital molecule from the simplest components, essentially from, basic carbon sources derived from your diet.
Amazing self-sufficiency.
But the true genius is in the regulation. The specific design of this pathway, particularly with HMG-CoA reductase as the master control, gives us a powerful lever for intervention, as we see so effectively with statin drugs. It's an elegant system of self-sufficiency and precise control.
That was an incredible journey through the biosynthesis. But simply making cholesterol isn't enough. The body needs to precisely control how much is made and when.
Right. Balance is key.
That brings us to section 3, the precision dial, the elaborate regulation of cholesterol synthesis.
Absolutely. The body has incredibly sophisticated mechanisms to maintain this delicate balance, preventing both deficiency and excess. As we just discussed, the master switch is the HMG-CoA reductase enzyme activity.
The main control point.
Its activity is directly inhibited by its immediate product, mevalonate, and perhaps most importantly by cholesterol itself, the final product of the entire pathway. That's classic feedback inhibition.
So if you're eating a lot of cholesterol, does your body's own internal production just naturally slow down? How responsive is that system in real time?
That's a great question, and it's a dynamic interplay. The source states that increased dietary cholesterol does indeed lead to a decrease in de novo synthesis, especially in the liver.
Okay.
It's a classic negative feedback loop. Your body senses sufficient levels, whether from internal production or diet, and intelligently adjusts production accordingly. It's not an off switch, but definitely a dial down.
It sounds like a complex thermostat, constantly adjusting production based on internal levels. But it's not just a simple feedback loop, is it? There are multiple layers of regulatory mechanisms involved.
Oh yeah, it's multi-layered.
Let's start with transcriptional control, how the body regulates the very gene expression for these enzymes.
This is where things get even more intricate. Cholesterol and its metabolites have the remarkable ability to directly repress the transcription of the mRNA for HMG-CoA reductase, meaning they reduce the instructions for making the enzyme.
How do they do that?
They do this by inhibiting a family of critical proteins called SREBPs, or sterile regulatory element binding proteins.
SREBPs, they sound important.
They absolutely are. SREBPs are critical transcription factors that regulate a wide range of genes involved in the cellular uptake and metabolism of not just cholesterol, but other lipids too.
Broader lipid metabolism.
Exactly. So when cholesterol levels are high, SREBPs are inhibited, which in turn reduces the production of the enzymes needed for cholesterol synthesis and uptake. The sources also point to INSIG, or insulin-induced gene, which is induced by insulin and resides in the endoplasmic reticulum membrane.
What's INSIG do?
INSIG actually inhibits SREP activation and promotes the degradation of HMG-CoA reductase, adding another layer of precise control. It basically helps keep SREBP anchored when cholesterol is present.
So it's like the body saying, we have enough, let's dial down the factory production lines and slow down imports.
That's a good way to put it.
Beyond gene expression, what about hormonal regulation?
Hormones play a significant role, signaling the body's overall metabolic state. For instance, insulin and thyroid hormone are known to increase HMG-CoA reductase activity, essentially signaling that energy is abundant and building blocks are available for synthesis.
Makes sense.
Conversely, hormones like glucagon, which signals low blood sugar, and glucocorticoids, which are involved in stress responses, decrease its activity, which makes sense when the body needs to conserve energy or respond to acute demands.
And then there's immediate control through post-translational modification, like flicking a switch on the existing enzyme itself.
Correct. That's for rapid adjustments. The activity of HMG-CoA reductase is reversibly modified through phosphorylation, adding a phosphate group, and dephosphorylation, removing it.
Phosphorylation usually turns things off, right?
In this case, yes. The sources highlight AMPK, or AMP-activated protein kinase, which phosphorylates and inactivates HMG-CoA reductase. Essentially, when cellular energy levels are low, high AMP levels, AMPK gets activated,
Sensing low energy.
leading to the inactivation of the enzyme and thus a rapid reduction in energy-consuming cholesterol synthesis. It's a quick way to conserve energy.
And this rapid mechanism ties back to the hormones, too.
Yes. Some of these phosphorylation mechanisms can be CANMP-dependent, making them immediately responsive to hormones like glucagon. Insulin, however, seems to have a dominant role in regulating this post-translational modification, often promoting the active dephosphorylated state of the enzyme.
Okay, so insulin pushes for synthesis when energy is available.
Generally, yes.
And finally, a subtle but interesting point the sources make. Diurnal variation. Cholesterol synthesis naturally fluctuates throughout the day.
Indeed. There's a natural daily rhythm where both cholesterol synthesis and reductase activity show predictable fluctuations, often peaking during the night when you're fasting.
Interesting.
This highlights the body's sophisticated internal clock influencing even molecular processes. All these layers of regulation, from genetic expression to direct enzyme modification, work together in a tightly integrated fashion to keep cholesterol production precisely tuned to the body's moment-to-moment needs. It's an incredibly intelligent system.
So we've explored how cholesterol is made and how its production is tightly regulated. But cholesterol isn't static. It's also constantly moving in and out of cells, being stored, and being used for various purposes.
Absolutely. It's always in flux.
This brings us to Section 4. Cellular balance, how your body meticulously keeps intracellular cholesterol levels in check.
This is truly a dynamic equilibrium, a constant balancing act between factors that increase cholesterol inside the cell and those that decrease it. The goal is to keep intracellular concentrations within a very narrow, healthy range, preventing both shortages and toxic overloads. Free cholesterol, especially, needs to be tightly controlled.
Let's look at the factors that increase intracellular cholesterol. I imagine one major way is simply taking it in from outside the cell, like receiving a delivery.
You're exactly right. One primary factor is the uptake of cholesterol-containing lipoproteins from the bloodstream. This happens via specific receptors on the cell surface, such as the famous LDL receptor, which we'll discuss, or other scavenger receptors like CD36.
Like docking stations.
Think of them as docking stations for cholesterol delivery vehicles, allowing cells to bring in what they need, primarily in the form of cholesterol esters.
And cells can also just absorb free cholesterol directly, right?
Yes, free cholesterol can be taken up directly from cholesterol-rich lipoproteins into the cell membrane itself. And of course, the de novo synthesis process we just meticulously explored is another major internal source of increased intracellular cholesterol.
All right, making it from scratch.
Finally, cholesterol can be liberated from storage within the cell. It's stored as cholesterol esters in lipid droplets, and when needed, these esters are hydrolyzed by the enzyme cholesterol ester hydrolase, releasing free cholesterol.
So that's how it gets in or is generated.
Yeah.
What about the factors that decrease intracellular cholesterol? How does the cell get rid of it or utilize it to maintain that balance?
One crucial pathway is the efflux of cholesterol from the cell membrane back out to HDL, those good cholesterol particles we often hear about.
Sending it back out.
Exactly. This is mediated by specific transporter proteins like ABCA1, ABCG1, or SRB1. It's a key part of what's called reverse cholesterol transport, essentially moving excess cholesterol out of cells and back towards disposal, primarily by the liver.
And then the cell can also prepare cholesterol for storage, effectively putting it into a molecular pantry for later use.
Precisely. It converts free cholesterol to cholesterol ester for storage, a process catalyzed by an enzyme called ASAT, or acetyl-CoA cholesterol acetyltransferase.
ACA.
This essentially packages cholesterol into a less reactive esterified form, preventing it from accumulating in its free form, which can be detrimental to the cell membrane structure and function.
So store it safely.
And finally, cells utilize cholesterol for synthesizing other vital steroids, such as hormones in steroidogenic tissues like the adrenal glands or gonads, or in the liver for producing bile acids, which is a major excretion pathway we'll get to.
It sounds like a very sophisticated internal inventory system. And at the heart of this system, particularly for how cells acquire cholesterol from the bloodstream, is the LDL receptor. The sources call it a gatekeeper for cellular cholesterol. How does it work its magic?
The LDL receptors are specialized proteins located on the cell surface, often concentrated in structures called clathrin-coated pits. These pits are like little indentations ready to engulf molecules.
Okay.
The receptor itself spans the cell membrane, and its specific binding region, known as the B100 binding region, is exposed on the outside of the cell, perfectly shaped to interact with the ApoB100 protein on LDL particles.
So LDL, the low-density lipoprotein, binds to this receptor. Then what happens?
Once an LDL particle binds, the entire complex, the LDL particle attached to its receptor, is taken up intact into the cell through a process called receptor-mediated endocytosis.
The cell swallows it whole.
Think of it that way, yes. Inside the cell, within organelles called lysosomes, the apoprotein components of LDL and the cholesterol ester are hydrolyzed, meaning they're broken down and liberated. The free cholesterol is then released into the cell's cytoplasm.
And the receptor.
Crucially, after this delivery, most of the LDL receptors are recycled back to the cell surface, ready to bind more LDL. It's an incredibly efficient recycling system.
That's very clever. And this influx of cholesterol acts as a critical signal for the cell, doesn't it? It's not just a delivery, it's also a message.
It's a fundamental signal for coordinated regulation. When a cell takes in a sufficient amount of cholesterol via the LDL receptor, it immediately triggers, well, several major responses.
Like what?
First, it actively inhibits the transcription of the genes that encode HMG-CoA synthase, and HMG-CoA reductase those key enzymes in cholesterol synthesis, and even the LDL receptor itself. This suppression happens via the SREBP pathway we mentioned earlier.
So less synthesis, fewer receptors.
Exactly. This ensures that if cholesterol is readily available from outside, the cell doesn't waste precious energy making its own or taking in too much more.
So it's like the cell saying, we have enough, slow down production, and stop bringing in more from outside.
That's a good analogy.
Yeah.
And the second response is that AC adductivity, the enzyme that esterifies cholesterol for storage, is stimulated.
Store the excess.
So the cell says, okay, we have enough free cholesterol right now, let's put some into storage for later.
Now, the sources introduce a relatively newer player in this whole LDL receptor story that's become a significant therapeutic target. PCSK9, or proprotein convertase subtilisin-kexin type 9. How does this protein fit into the picture?
PCSK9 plays a direct and critical role in regulating the lifespan and recycling of the LDL receptor. Normally, after delivering its cargo, the LDL receptor returns to the cell surface.
Right, the recycling part.
However, when PCSK9 binds to the LDL receptor, it prevents this recycling. Instead, it targets the LDL receptor for degradation within the cell, sending it to the lysosome along with the LDL.
So it destroys a receptor.
Essentially, yes. So if PCSK9 is highly active, fewer LDL receptors return to the surface, meaning the cells can't clear LDL from the blood as effectively. This leads directly to higher blood cholesterol levels.
Which means if you can inhibit PCSK9, you can essentially keep more LDL receptors active on the cell surface, leading to lower blood cholesterol.
Exactly.
That's a profound insight for drug development, offering a new pathway for intervention.
Precisely. The overall goal of all these complex, interconnected mechanisms, the influx and efflux, the synthesis and storage, the LDL receptor, and even PCSK9, is to ensure that the free cholesterol content of the cell is kept within relatively narrow optimal limits. This ensures the cell can respond dynamically to its demand for membranes, for steroid hormones, or for bile acid synthesis without being overwhelmed or starved of this vital lipid. It's a masterclass in biological precision.
That was a brilliant breakdown of cellular cholesterol balance. But cholesterol isn't confined to individual cells. It's constantly on the move throughout your entire body.
Yeah, it's got to get around.
So let's turn our attention to Section 5. The arterial network cholesterol transport in plasma lipoproteins.
Think of your plasma, your blood, as a vast, watery highway. Cholesterol is a lipid, meaning it's inherently oily and doesn't mix well with water.
Like oil and water.
Exactly. So it needs specialized vehicles for transport. It's primarily transported in plasma in the form of cholesterol ester, which is its more stable storage and transport form,
Yeah.
all encapsulated within these sophisticated particles called plasma lipoproteins.
And these are the famous players we often hear about: chylomicrons, VLDL, LDL, and HDL.
Exactly. Each has a specific role in this transport network, like different types of cargo ships or delivery trucks. Our sources emphasize that in humans, the highest proportion of cholesterol is actually found in LDL.
Which is why LDL gets labeled bad.
That's generally why, yes.
Yeah.
And it's a dynamic system. Dietary cholesterol equilibrates with plasma cholesterol in a matter of days, and with tissue cholesterol in weeks, showing that there's a constant active exchange and balancing act going on.
So let's trace the journey of dietary cholesterol. What happens to that egg yolk or piece of meat once we eat it? Where does it all begin?
Okay, when you consume dietary cholesterol ester, it's first hydrolyzed in your gut to free cholesterol. This free cholesterol, along with any unesterified cholesterol already present in your diet and other lipids like triglycerides, is then absorbed by the intestinal cells.
Into the intestinal cells.
Once absorbed, this cholesterol, combined with cholesterol synthesized in the intestines themselves, gets incorporated into large triglyceride-rich lipoprotein particles called chylomicrons.
So chylomicrons are the immediate transporters for the cholesterol we eat. Where do they take their cargo?
The chylomicrons are too large to directly enter your bloodstream capillaries, so they first travel through the lymphatic system and then eventually enter the bloodstream via the thoracic duct.
Okay, through the lymph first.
Right. Then, in the bloodstream, they interact with lipoprotein lipase on capillary walls, which breaks down their triglycerides. A remarkable 95% of the cholesterol carried by chylomicrons is ultimately delivered to the liver in the form of chylomicron remnants, after much of their triglyceride content has been delivered to other tissues for energy or storage.
So the liver is the main destination for dietary cholesterol remnants. What happens next? How does the liver distribute cholesterol to the rest of the body for its various needs?
That brings us to hepatic export and peripheral delivery. The liver then packages and secretes most of its cholesterol, along with newly synthesized triglycerides in VLDL, or very low-density lipoprotein, another type of triglyceride-rich particle.
Okay, VLDL leaves the liver.
As VLDL travels through the bloodstream, it also interacts with lipoprotein lipase, gradually unloading its triglycerides to various tissues. As it does this, it undergoes a transformation, first becoming IDL, or intermediate density lipoprotein. And ultimately, after losing more triglycerides and becoming relatively enriched in cholesterol, it becomes LDL, low-density lipoprotein. Much of the cholesterol that was originally in the VLDL is retained during this entire transformation process.
And then the LDL particles, now rich in cholesterol, deliver their cargo to other cells throughout the body.
Precisely. LDL is then taken up by the LDL receptor, which we just discussed, not just in the liver, but also in extrahepatic tissues. These are all the tissues outside the liver that need cholesterol for membrane synthesis, hormone production, and so on.
And this is why LDL is often referred to as bad cholesterol.
Because it's the primary vehicle delivering cholesterol to tissues, and if there's too much LDL circulating or the receptors aren't working well, it can contribute to buildup in artery walls.
Which brings us to the good cholesterol, HDL, and the fascinating process of reverse cholesterol transport. How does HDL play its protective role, especially in the context of atherosclerosis?
HDL's primary function in reverse cholesterol transport is, like you said, protective. It removes excess cholesterol from peripheral tissues, like the artery walls where plaques might form, and transports it back to the liver for excretion.
How does it do that?
A key enzyme in this process is LCAT, or lecithin, cholesterol acyl transferase.
LCAT, the esterifier, what does it do?
LCAT activity is tightly associated with HDL, specifically with HDL particles containing apolipoprotein AI, which activates it. As free cholesterol from cells and tissues is taken up by nascent discoidal HDL, LCAT esterifies it, meaning it attaches a fatty acid to it, forming cholesterol ester.
Okay.
This newly formed, more hydrophobic cholesterol ester then moves from the surface to the core of the HDL particle, causing the HDL to become spherical and mature.
So it's like a molecular vacuum cleaner, right?
Okay.
As cholesterol gets esterified and moves into the core, it creates a concentration gradient that continuously draws more free cholesterol out of the tissues.
That's a perfect analogy. This continuous process enables HDL to effectively function in reverse cholesterol transport, essentially vacuuming up excess free cholesterol from peripheral cells and even from the surface of other lipoproteins.
And then there's another protein, CETP, or cholesterol ester transfer protein. What role does it play in this complex exchange?
CETP is associated with HDL in plasma. Its crucial role is to facilitate the transfer of cholesterol ester from HDL to VLDL, IDL, and LDL.
So it moves the cholesterol ester off HDL.
It does, and in exchange, it receives triacylglycerol from those lipoproteins onto HDL. It effectively swaps cholesterol esters for triglycerides between these lipoproteins.
Look at that.
This exchange mechanism serves two important purposes. One, it helps relieve product inhibition of LCAT activity in HDL, allowing LCAT to continue esterifying cholesterol effectively.
Keeps the vacuum running.
Kinda, yeah. And two, it provides a route for that cholesterol ester to get back to the liver.
And where does all this exchanged cholesterol ultimately end up?
In humans, much of the cholesterol ester that was originally formed by LCAT on HEL and then exchanged by CETP eventually finds its way to the liver via the uptake of VLDL remnants, which are IDL, or via LDL uptake through the LDL receptor.
So it completes the circle back to the liver.
Exactly. This effectively completes the reverse transport pathway, ensuring that excess cholesterol from the periphery makes its way back to the liver, where it can be processed for excretion, often as bile acids. Additionally, the triacylglycerol-enriched HDL2 can also directly deliver its cholesterol to the liver via other receptors like SRB1 as part of the ongoing HDL cycle. It's an incredibly efficient and interconnected system designed to precisely manage cholesterol levels throughout the body, acting as a critical surveillance and cleanup crew.
So cholesterol gets made, it gets transported, but eventually any excess needs to leave the body.
Right, you can't just keep accumulating it.
That brings us to section 6. The exit strategy, cholesterol excretion, and bile acids.
This is a critical piece of the puzzle, as excretion is the final essential step in maintaining that delicate cholesterol balance within your body. Cholesterol is primarily eliminated from the body via the bile, which is a digestive fluid produced by your liver.
Okay, through the bile.
It can be excreted in two primary forms, either as unesterified free cholesterol dissolved in the bile with the help of bile salts and phospholipids, or, much more significantly, after it's been chemically converted into bile acids in the liver. This conversion accounts for a major portion of cholesterol elimination.
And what happens once it's in the bile and then passes through the intestines? Does it just get flushed out?
Some of the free cholesterol excreted in bile might be reabsorbed, but a portion is lost in feces. Also, as it moves through your lower intestine, intestinal bacteria act on any remaining unabsorbed cholesterol. The principal sterol that ends up in your feces is a molecule called coprostanol, which is essentially a modified form of cholesterol produced by these gut bacteria.
So bile acid synthesis is a major excretory route for cholesterol. How exactly are these bile acids formed from cholesterol in the liver?
The primary bile acids are synthesized in the liver directly from cholesterol through a series of enzymatic steps. The two key examples found in most mammals, including humans, are cholic acid and chenodeoxycholic acid. Cholic acid is generally the most abundant.
And there's a principal regulatory step here, just like in cholesterol synthesis, isn't there? A key control point.
Absolutely. The 7-hydroxylation of cholesterol is considered the first and most important regulatory step in acid biosynthesis. This specific reaction is catalyzed by an enzyme called cholesterol 7-hydroxylase, also known as CYP7A1.
CYP7A1.
Right. It's a microsomal cytochrome P450 enzyme. It's the rate-limiting enzyme, meaning it determines the overall speed of the pathway. Like other monooxygenases, it requires oxygen, NADPH, and cytochrome P450 for its activity. Subsequent hydroxylation steps in the pathway are also catalyzed by other monooxygenases.
So CYP7A1 is the gatekeeper for converting cholesterol into bile acids. And the sources mention divergent pathways and even a second pathway for bile acid synthesis.
Yes. Early in the bile acid biosynthesis pathway, it actually divides into two main subpathways. One leading to acetyl-CoA, which is characterized by an extra hydroxyl group on position 12, and another leading to chenodeoxycholic-CoA.
Okay. Additionally,
a significant proportion of primary bile acids are also synthesized via a distinct mitochondrial pathway. This alternative route begins with a 27-hydroxylation of cholesterol, catalyzed by cytochrome P450 sterol 27-hydroxylase, or CYP27A1 as its first step. So the liver has a couple of different ways to get the job done, providing some flexibility.
And once these primary bile acids are formed, they undergo another crucial modification before entering the bile, right? They're not just secreted as is.
Correct. They are conjugated. Primary bile acids enter the bile as glycine or taurine conjugates. This conjugation typically takes place in cellular compartments called liver peroxisomes.
Why conjugate them?
Conjugation makes them more water-soluble and better detergents for fat digestion. In humans, the ratio of glycine to taurine conjugates is usually about 3.1. Once they're in the alkaline environment of the bile with a pH of 7.6 to 8.4, these conjugates are commonly referred to as bile salts, and they're crucial for emulsifying fats during digestion.
Now, things get even more interesting when these bile acids reach the intestine. The sources mention secondary bile acids and the crucial role of our gut bacteria.
Indeed. The primary bile acids that enter the intestine via the bile are not simply absorbed unchanged. They are further metabolized by the vast activity of the intestinal bacteria living in your gut.
Our gut microbiome at work again.
Exactly. These bacteria perform chemical transformations like deconjugation, removing the glycine or taurine, and 7-dehydroxylation, which removes a hydroxyl group. This leads to the production of what we call secondary bile acids, the most notable examples being deoxycholic acid from cholic acid and lithocholic acid from chenodeoxycholic acid.
This sounds like a lot of bile acids are being produced, modified, and used. Does it all just get excreted, or is there a clever recycling system at play?
This leads us to one of the most remarkably efficient recycling systems in the body, the enterohepatic circulation.
Enterohepatic circulation. Okay.
While most products of fat digestion, including cholesterol, are absorbed relatively early in the small intestine, the primary and secondary bile acids are absorbed almost exclusively in the ileum, which is the very last part of the small intestine.
So they travel quite a distance before being reabsorbed. How much actually makes it back to the liver?
An impressive 98% to 99% of these bile acids are reabsorbed and returned to the liver via the portal circulation, a specialized blood vessel system connecting the gut to the liver.
Wow, 98, 99% recycled.
It's highly efficient. This recycling process is what we call the enterohepatic circulation. There's a notable exception, though. Lithocholic acid, due to its low solubility, isn't reabsorbed to any significant extent and is mostly excreted in feces.
So only a tiny fraction of the total bile acid pool is actually lost to the feces with each cycle. But the sources note that this small fraction still represents a major pathway for cholesterol elimination. How can that be if so little is lost each time?
Because the entire bile acid pool, which is relatively small, about 3 to 5 grams, is cycled through the intestine a remarkable 6 to 10 times daily.
6 to 10 times a day.
Yeah. So even though only a tiny fraction is lost with each cycle over the course of a day, the cumulative amount eliminated in feces represents a substantial amount of cholesterol removal from the body. To maintain a constant pool size, an equivalent amount of bile acid to what is lost is synthesized from cholesterol each day by the liver.
Keeping the balance.
This constant turnover and replenishment is managed by a sophisticated system of feedback controls.
And how is that pool size precisely maintained through regulation? Is it back to that crucial CYP7A1 enzyme again?
Exactly. The CYP7A1 reaction, the 7-hydroxylation of cholesterol, remains the principal rate-limiting step in bile acid biosynthesis. The activity of this enzyme is feedback regulated by a nuclear bile acid binding receptor called the farnesoid X receptor, or FXR.
FXR. How does it work in this feedback loop?
When the size of the bile acid pool returning to the liver via the enterohepatic circulation increases, signaling that there's plenty of bile acid around, FXR is activated by binding to these bile acids.
Okay.
Once activated, FXR then effectively suppresses the transcription of the CYP7A1 gene. This reduces the synthesis of the rate-limiting enzyme, thereby slowing down new bile acid production. The sources specifically mention that chenodeoxycholic acid is particularly potent in activating FXR.
So it's a very precise way for the body to say, we have enough bile acids in circulation, so let's dial down production.
Yeah.
Are there other factors influencing CYP7A1 activity beyond FXR?
Yes, CYP7A1 activity is also enhanced by cholesterol itself, whether it's of endogenous origin from your body's own synthesis or from dietary intake. This provides another direct link between overall cholesterol levels and its excretion pathway. More cholesterol substrate can drive the process.
Makes sense.
And it's also regulated by various hormones, including insulin, glucagon, glucocorticoids, and thyroid hormone, highlighting the extensive hormonal influence on metabolic pathways, integrating bile acid synthesis into the body's broader metabolic state.
We've delved deep into the molecular mechanisms of cholesterol synthesis, transport, and excretion. Now let's bring it all together and talk about what all this intricate biochemistry means for our real-world health, the crucial clinical relevance of cholesterol.
This is truly where the rubber meets the road, as they say. The direct link we need to understand is between serum cholesterol levels and the incidence of atherosclerosis and coronary heart disease, or CHD.
Atherosclerosis.
It's fundamentally an inflammatory disease characterized by the deposition of cholesterol and cholesterol ester from plasma lipoproteins, particularly LDL, into the artery wall, forming plaques. It is unequivocally a leading cause of heart disease worldwide.
And what are the key risk factors related to cholesterol levels that we should be aware of?
The sources highlight that elevated plasma cholesterol levels, specifically total cholesterol greater than 5.2 millimole or roughly 200 milli-GDL, are one of the most important factors promoting atherosclerosis. Elevated LDL cholesterol is particularly implicated.
And triglycerides.
It's now also recognized that elevated blood triacylglycerol levels are an independent risk factor, meaning they contribute to risk even if cholesterol levels aren't extremely high, often reflecting issues with VLDL or remnant metabolism.
So it's not just cholesterol, but also the form of fats circulating. What about other conditions that might predispose someone to accelerated atherosclerosis?
Diseases where there's a prolonged elevation of VLDL, IDL, chylomicron remnants, or LDL in the blood conditions, like uncontrolled diabetes, mellitus, lipid nephrosis, a kidney condition, hypothyroidism, and other forms of inherited hyperlipidemia are very often accompanied by premature or more severe atherosclerosis.
Because the balance is off.
The body's balance of these lipoproteins is crucial, yes.
And then there's HDL, the good cholesterol. The sources mention a very important inverse relationship with coronary heart disease.
Correct. There's a strong inverse relationship between HDL concentrations, specifically a subfraction called HDL2, and coronary heart disease risk. Higher HDL is generally protective.
Why is that?
This is precisely why the LDL-HDL cholesterol ratio is considered a good predictive parameter for cardiovascular risk. It directly aligns with HDL's protective function in reverse cholesterol transport, effectively moving excess cholesterol out of artery walls and back to the liver for disposal.
Right, the cleanup crew.
Exactly. It's also worth noting that humans are one of the few species where diets high in cholesterol and saturated fat can actually induce this disease, unlike many other animals.
So diet clearly plays an important role. How much can we really influence our serum cholesterol through what we eat, given that our bodies make so much of it internally?
That's a key question. Hereditary factors actually play the most important role in determining an individual's long-term serum cholesterol concentrations.
So genetics is primary.
It plays a huge role. However, dietary and environmental factors are undeniably significant, and this is where we have considerable influence. The most beneficial dietary change, according to our sources, is substituting polyunsaturated and monounsaturated fatty acids for saturated fatty acids.
Trading fats. What are some good practical sources of these beneficial fats we should be looking for?
Plant oils like corn oil and sunflower seed oil are high in omega-6 polyunsaturated fatty acids. Olive oil is renowned for its high concentration of monounsaturated fatty acids. And fish oils, rich in omega-3 fatty acids, are also considered highly beneficial for overall heart health.
And what exactly do these beneficial fats do at a molecular level that helps lower blood cholesterol or improve the profile?
One key mechanism by which unsaturated fatty acids contribute to lower blood cholesterol is by upregulating LDL receptors on the cell surface, particularly in the liver.
More receptors means?
More LDL can be cleared from the blood, making it less atherogenic. Additionally, omega-3 fatty acids specifically have powerful anti-inflammatory effects and can significantly lower triacylglycerol concentrations, further contributing to their overall cardiovascular benefits.
And what about the less beneficial or even harmful dietary factors? What should we try to limit?
Saturated fatty acids, found in things like butterfat, beef fat, coconut oil, and palm oil, are generally less beneficial for lipid profiles, often raising LDL cholesterol. They tend to promote the formation of smaller VLDL particles that contain relatively more cholesterol. These particles are then utilized by extrahepatic tissues at a slower rate than larger particles tendencies regarded as atherogenic. Furthermore, the sources highlight that sucrose and fructose, common sugars, have a greater effect in raising blood lipids, particularly triacylglycerols, compared to other carbohydrates like starch.
So high sugar intake impacts triglycerides.
Significantly, yes. So it's not just about fat type, but also sugar intake.
Beyond diet, what about general lifestyle factors? What else impacts our cholesterol and heart health?
A wide range of lifestyle factors contribute to the risk of coronary heart disease. These include high blood pressure, smoking, male gender, obesity, especially abdominal obesity, lack of regular exercise, and surprisingly, even drinking soft water as opposed to hard water, though the mechanism there is less clear.
Are there any other more subtle influences on cholesterol levels that might surprise us?
Yes. Factors associated with elevated plasma-free fatty acids, such as chronic emotional stress and even coffee drinking in some individuals, likely related to unfiltered coffee, can lead to an increased output of triacylglycerol and cholesterol into the circulation in VLDL from the liver.
Stress and coffee. Interesting.
On the protective side, premenopausal women appear to be protected against many of these deleterious factors, which is thought to be related to the beneficial effects of estrogen on lipid profiles and vascular function.
And moderate alcohol consumption is often cited as potentially beneficial, particularly red wine. What's the science there?
That's right. Moderate alcohol consumption is associated with a lower incidence of coronary heart disease, possibly due to an elevation of HDL concentrations.
Raises the good cholesterol.
It can, possibly, by increasing synthesis of apolipoprotein AI, a key component of HDL, and perhaps changes in the activity of cholesterol ester transfer protein, CETP.
Red wine is specifically claimed to be particularly beneficial, perhaps because of its antioxidant content, like resveratrol, though the evidence is complex. Moderation being key, presumably.
Absolutely. And importantly, regular exercise significantly impacts lipid profiles for the better. It consistently lowers plasma LDL, raises HDL, and reduces triacylglycerol concentrations, most likely due to increased insulin sensitivity, which enhances the expression and activity of lipoprotein lipase.
So, diet and lifestyle are foundational, but sometimes they aren't enough, or genetics play too strong a hand.
Exactly.
That's where pharmacological interventions come in. What are the key drug classes used to manage cholesterol when lifestyle changes aren't sufficient?
The most widely known and highly effective class are the statins. They've proven incredibly efficacious in lowering plasma cholesterol, specifically LDL cholesterol, and profoundly preventing heart disease events.
And how do they work, given our deep dive into the biosynthesis pathway?
As we discussed earlier, statins act by directly inhibiting HMG-CoA reductase, which is the rate-limiting enzyme in cholesterol synthesis within the liver and other cells.
Blocking the fat stream.
Essentially, yes. By doing so, they reduce the de novo production of cholesterol within the liver. Crucially, this reduction in internal cholesterol also signals the liver to upregulate or increase the number of LDL receptors on its surface.
Ah, so a double effect.
Exactly. More receptors means more LDL is cleared from the blood. Common examples you might recognize include atorvastatin, simvastatin, fluvastatin, and pravastatin.
What about other approaches beyond just inhibiting synthesis?
Another drug, ezetimibe, reduces blood cholesterol levels by inhibiting cholesterol absorption in the intestine. It does this by specifically blocking a protein called Niemann-Pick C-like 1 and PC1L1, which is essential for cholesterol uptake from the gut into intestinal cells.
Blocks absorption.
Right. Then there are fibrates, such as clofibrate, gemfibrozil, and fenofibrate. These mainly work to lower plasma triacylglycerols by decreasing the liver secretion of VLDL, which contains both triacylglycerol and cholesterol, and also by increasing LPL activity.
Fibrates for triglycerides mainly.
Generally, yes, though they can have modest effects on LDL and HDL too. Nicotinic acid, or niacin, also lowers triglycerides and VLDL and notably raises HDL, but its use is often limited by side effects like flushing.
And then the newer class of drugs related to PCSK9, which we touched on earlier, these seem to be quite revolutionary.
They truly are. The PCSK9 inhibitors are designed with a very targeted rationale. Since PCSK9 reduces the number of LDL receptors exposed on the cell membrane, thereby raising blood cholesterol levels, inhibiting its activity would be powerfully anti-atherogenic.
Stops the receptor breakdown.
Precisely. These drugs, usually monoclonal antibodies, prevent PCSK9 from binding to and degrading LDL receptors. This leads to significantly more receptors remaining on the cell surface and thus dramatically increased clearance of LDL from the bloodstream, leading to very significant LDL lowering.
Powerful stuff.
Two such compounds, alirocumab and evolocumab, have been approved, and others are in development, marking a significant advancement in lipid-lowering therapies, especially for those with familial hypercholesterolemia or those who don't reach goals with statins alone.
That was a comprehensive look at how we manage cholesterol through diet, lifestyle, and medication. But sometimes, despite our best efforts, or even without any clear lifestyle factors, cholesterol levels can be abnormal due to our genes.
That's a really important point. Genetics plays a major role.
This brings us to Section 8, Inherited Variations Understanding Dyslipoproteinemias.
That's right. These are disorders where there's an inherited defect in lipoprotein metabolism. They're specifically called primary dyslipoproteinemias, It's important to distinguish them from secondary abnormalities.
Which are caused by other diseases.
Exactly. Conditions like diabetes mellitus, hypothyroidism, or kidney disease can also cause abnormal lipoprotein patterns that resemble the inherited conditions. For our deep dive, we'll focus on some of the key primary inherited conditions, highlighting what goes wrong and why it matters.
Let's start with hypolipoproteinemias, conditions where lipoprotein levels are abnormally low. The first one the sources mention is abetalipoproteinemia. What happens here?
In abetalipoproteinemia, there's a fundamental defect in loading ApoB with lipid, specifically involving a protein called MTP, microsomal triglyceride transfer protein. This is critical because ApoB is essential for the formation of chylomicrons, VLDL, and LDL.
So no ApoB lipoproteins are made.
Essentially, yes. The consequence is that none of these lipoproteins are formed or secreted. This leads to very low blood acetylglycerols and cholesterol, but paradoxically also significant intestinal malabsorption because fats can't be properly packaged and transported.
What are the effects?
It leads to an accumulation of fats in the intestine and liver and severe deficiencies of fat-soluble vitamins. Without intervention, which now involves high doses of fat-soluble vitamins, especially vitamin E and A, it could lead to severe neurological problems and early death.
And then there's a group of conditions involving HDL, familial alpha-lipoprotein deficiency, which includes Tangier disease, fish-eye disease, and ApoAI deficiencies. What are the common threads here?
These conditions are all characterized by strikingly low or even near absence of HDL. Tangier disease, for example, involves defects in the ABCA1 transporter, which is crucial for cholesterol efflux to HDL.
The first step in reverse transport.
Exactly. This can lead to hypertriacylglycerolemia, meaning high levels of triglycerides in the blood, often due to inactive lipoprotein lipase, LPL, because ApoA, deficient here, also plays a role. While LDL levels might be low, cholesterol esters accumulate in tissues like tonsils, making them orange. Nerves and arteries and atherosclerosis can still develop later in life, underscoring the protective role of HDL. Fish-eye disease involves LSAT deficiency.
Now let's move to the hyperlipoproteinemia's conditions with abnormally high lipoprotein levels.
Yep.
The first is familial lipoprotein lipase deficiency, or type Y.
In familial lipoprotein lipase deficiency, type I, there's severe hypertriacylglycerolemia, really high triglycerides due to either a deficiency of LPL itself, an abnormal LPL, or a deficiency in apolipoprotein CI, which is required to activate LPL.
So triglycerides can't be broken down properly.
Right. This results in very slow clearance of chylomicrons and VLDL from the blood, leading to a milky appearance of the plasma, lipemia. Interestingly, despite the very high triglycerides, there isn't usually an increased risk of coronary disease in this specific condition, though patients are at high risk for recurrent, painful pancreatitis.
And then the big one, the one that often makes headlines, and it is a significant clinical challenge. Familial hypercholesterolemia, type DIA. This sounds like a critical example.
It truly is. Familial hypercholesterolemia, FH, is characterized by severely elevated LDL levels and hypercholesterolemia right from birth, and it's strongly associated with premature atherosclerosis and coronary heart disease, often presenting in the 30s or 40s, sometimes even earlier in severe forms.
What's the genetic cause?
The defect is most often in the gene for the LDL receptor itself. Mutations mean the receptor doesn't work properly or isn't made in sufficient numbers. So LDL is not effectively cleared from the blood and just keeps circulating at high levels. Less commonly, it can be due to a mutation in the apolipoprotein B100 ligand region, the part of LDL that the receptor binds to, or even mutations in PCSK9 causing it to be overactive.
So multiple ways to break the system.
Either way, the result is too much LDL circulating and being available to deposit in artery walls, leading to early heart disease.
That explains why it's such a significant risk factor for early heart disease. What about familial type 3 hyperlipoproteinemia? The sources call it broad beta disease or remnant removal disease.
In familial type 3 hyperlipoproteinemia or dysbeta-lipoproteinemia, the core issue is a deficiency in the efficient clearance of remnants, chylomicron remnants, and VLDL remnants, IDL, by the liver.
Why can't the liver clear them?
It's typically due to an abnormality in apolipoprotein E. Patients usually lack the common E3 and E4 isoforms of ApoE and possess only the E2 isoform, which doesn't bind well to the remnant receptors, including the LDL receptor, on liver cells.
So the remnants build up.
Exactly. This leads to a buildup of these remnants in the plasma, causing elevations in both cholesterol and triglycerides. characteristic fatty deposits called xanthomas, especially pulmonar xanthomas, and indeed accelerated peripheral and coronary atherosclerosis.
Then there's familial hypertriacyclic or type 5.
This condition primarily involves the overproduction of VLDL by the liver, leading to high triglyceride levels. It's often associated with other metabolic imbalances like glucose intolerance, insulin resistance, obesity, and hyperinsulinemia.
How does cholesterol fit in?
Cholesterol levels will rise in conjunction with the VLDL concentration, but LDL is often normal or low, and HDL is often low. While sometimes considered benign, this pattern is commonly linked to coronary heart disease, type 2 diabetes mellitus, obesity, and even alcoholism can exacerbate it.
And on the flip side of the coin, there's familial hyperalpha-lipoproteinemia. This sounds like a good problem to have.
It definitely seems to be. This is a rare and rather unique condition characterized by increased HDL concentrations, sometimes due to genetic factors like variations in CETP activity. It's seemingly beneficial for health and longevity, illustrating that not all genetic variations in lipoprotein metabolism are harmful. Some can be protective.
What about hepatic lipase deficiency?
Hepatic lipase is an enzyme primarily on liver cells that helps process remnants and HDL. In hepatic lipase deficiency, this enzyme is deficient. This leads to an accumulation of large triacylglycerol-rich HDL, HDL2-like particles, and VLDL remnants in the bloodstream, contributing to both xanthomas, the fatty deposits, and potentially coronary heart disease.
And familial lecithin-cholesterol acetyltransferase, or LCAT, deficiency. It sounds like it would directly impact the reverse cholesterol transport we just discussed.
It does, profoundly. The absence of LCAT completely blocks the normal process of esterifying cholesterol and HDL, which is essential for HDL maturation and reverse cholesterol transport.
What happens to HDL?
As a result, HDL remains in its nascent discoidal form and is incapable of taking up and esterifying cholesterol effectively. This leads to very low plasma concentrations of cholesterol esters because LCAT isn't making them. Free cholesterol accumulates in tissues, leading to corneal opacities, kidney disease, and anemia. An abnormal LDL fraction, often called lipoprotein X, which is also seen in patients with cholestasis, bile flow blockage, and abnormal VLDL are also present.
Finally, familial lipoprotein excess. What exactly is LPA and why is its excess harmful for the heart?
LPA, or lipoprotein A, consists of one molecule of LDL attached via a disulfide bond to one molecule of a unique protein called apolipoprotein, or APO.
And apoP is the issue.
What's particularly concerning is that apolipoprotein A shows striking structural similarities to plasminogen, a protein involved in breaking down blood clots, fibrinolysis. Because of this similarity, elevated LPA levels, which are largely genetically determined, are linked to premature coronary heart disease. It's thought to contribute both to atherosclerosis, like LDL, but also importantly to thrombosis, clot formation, through its potential to interfere with and inhibit fibrinolysis. So it contributes to both plaque buildup and potentially dangerous clot formation within those plaques.
So understanding these inherited variations is crucial for both accurate diagnosis and highly targeted treatment, revealing just how deeply our genes influence our lipid profiles and consequently our cardiovascular health. It's a complex interplay.
Absolutely. It highlights why personalized approaches are becoming so important in managing lipid disorders.
Well, we've truly taken a deep dive today into the multifaceted world of cholesterol.
We certainly covered a lot of ground.
From its absolutely essential life-sustaining roles to its potential to cause severe disease, we've traced its intricate journey through your body.
We started from the simplest atomic building blocks, acetyl-CoA, followed its complex synthesis into a vital molecule, tracked its intricate regulatory pathways involving enzymes like HMG-CoA reductase and feedback loops,
and then observed its transport across the vast network of your circulatory system, understanding the roles of each key player, chylomicrons, VLDL, LDL, HDL, and all the associated enzymes and proteins like LCAT and CETP.
And we didn't stop there. We uncovered how your body finally gets rid of cholesterol, mainly through bile acid synthesis controlled by enzymes like CYP7A1 and receptors like FXR.
And connected all of these sophisticated biochemical processes to real-world health implications, exploring how diet, lifestyle, cutting-edge drugs like statins and PCSK9 inhibitors, and even your unique genetic makeup play a part in this delicate balance.
You now hopefully have a comprehensive, almost mastery-level understanding of how your body manages this crucial lipid.
As you reflect on all this information, consider the remarkable balance your body constantly strives to maintain.
Every single mechanism we discussed, from enzyme activity being switched on and off to receptor recycling and intricate transport systems, is a testament to this constant, tireless effort to keep you healthy. It's truly elegant biochemistry in action.
So how does understanding the detailed mechanisms of cholesterol synthesis and transport and the specific roles of different lipoproteins change your perspective on everyday health choices and medical advice?
Yeah, maybe you look at that lipid panel from your doctor a little differently now.
What further questions does this deep dive spark for you about the profound interconnectedness of biochemical processes and their ultimate impact on your well-being? Maybe thinking about how inflammation ties into atherosclerosis or the role of the gut microbiome beyond just secondary bile acids.
We encourage you to keep exploring, keep questioning, and keep applying this knowledge to become truly well-informed in your own health journey. Understanding the why can be incredibly empowering.
Until next time, keep diving deep.